Thank you so much for this very informative case-based discussion on respiratory syndromic testing. Begin with this session. Good afternoon, ladies and gentlemen. I am Dr. Priya, your online moderator for this session. I welcome you all to this post-lunch guest lecture session. Through this session, we are going to uh, explore the various. We are going to explore the very important topic on basic COVID-19 virology, which is a very important topic in this current COVID scenario. Before proceeding, I would like to talk about the question answer session. This question answer session will be taken at the end of the talk. You all are requested to keep posting your queries and question in the chat box.
I'll quickly run through the slides.
I would like to take this opportunity to introduce the chairperson of this session. Uh, it is an honor and privilege for me to introduce the chairperson, Dr. Anita Chakravarti ma'am. Can you please allow me to share the screen? Yeah, okay. Thank you. Thank you. Dr. Anita Chakravarti ma'am is currently working as a professor of microbiology at North TMC Medical College, Hindu Rao Hospital, New Delhi. Ma'am is the ex-director professor and head of the department in the Department of Microbiology, Maulana Azad Medical College. She has a vast academic experience of more than 40 years, and she has more than 130 publications in various high-impact national and international journals and various book chapters to her credit. Her area of interest is virology. She has planned and set up the Advanced Virology Laboratory at Maulana Azad Medical College. Her research are mostly focused on standardization and application of molecular technique for diagnosis and understanding the immunopathogenesis of dengue virus, hepatitis C, and chikungunya virus. Her data from various scientific studies on rubella virus in this community has led to early incorporation of this rubella vaccine in the universal immunization program in the Delhi government. She has been investigated to various research projects which are funded by DST, ICMR, and many more. She is an expert member of committees for BIRAC, ICMR, and many more. She is bestowed with many national and international fellowships, awards, and honors, like the State Award for Government of NCT, ICMR National Award, the JB Srivastava National Award, Indian Association of Medical Microbiologists Endowment Oration Award, Dr. S. Radhakrishnan Memorial National Teaching Award. She was also awarded the WHO Fellowship and UGC Fellowship under Indo-Hungarian Cultural Exchange Program. I welcome you, ma'am, to this session, and I would request, ma'am, to kindly begin the session by introducing the speaker of this session, Dr. V. Ravi, sir. Thank you so much, ma'am. Over to you.
Thank you, Pragna, for your kind words. And I welcome all the delegates to this interesting session after a delicious virtual lunch. I thank the organizers for inviting me for this session. And it is indeed a pleasure introducing Dr. Ravi, who's a very eminent virologist. And he actually doesn't need any introduction. And he was a senior professor, head of the Department of Neurovirology. And he was also the dean of the academic branch, as well as the registrar for a very long time. He is passionate about the viruses. His dedication to the viruses is very well known, and we can call him a hardcore virologist who is running after the crimes of the virus and trying to have his own solutions to them. So, Dr. Ravi, I invite you to present the guest lecture on the basic COVID virology.
Thank you, Dr. Anita, for a very kind introduction. And good afternoon to everybody. At the outset, let me thank the organizers for giving me this opportunity to share some information on the basic virology of SARS-CoV-2. I just pull up my screen. I'm going to talk to you on SARS-CoV-2 virology. Can I go to the next slide, please? Somebody is operating the slides. Okay, in the next 20 minutes, I try to cover some major aspects on history, classification, structure, genomic organization, physical chemical properties, basic biology, a word on the pathogenesis and therapeutic approaches. Next slide, please. If you look at the history of human coronaviruses, they've been known for the last 60 years. The first human coronavirus was human coronavirus 229E, followed by NL63, and OC43 and HKU1, which was discovered in 2004. These viruses usually cause mild upper respiratory illnesses and gastrointestinal symptoms. The first severe uh, coronavirus known as SARS-CoV emerged in 2003. All of us know about it. And then we had the Middle Eastern respiratory virus in 2012, and last year, the novel coronavirus SARS-CoV-2. Next slide, please. They are enveloped viruses. Their genetic material is RNA. They are distributed broadly among humans, mammals, and birds. And most important is they exhibit genetic diversity, and they also cross over from one species to another, which is called spillover events. The name corona is derived because of, under the electron microscopy, it has a crown-like appearance, so it's called coronaviruses. Next slide, please. If you look at the spillover events, next slide, please. Can I have the next slide, please? There have been spillover events periodically occurring in the last 60 years, leading to emergence of coronaviruses, especially the severe acute respiratory syndrome viruses. Bats and rodents are the natural hosts, reservoirs, so to say, of coronaviruses. The severe acute respiratory syndrome and MERS coronaviruses, the reservoir is bats. There is an intermediate host where the spillover first occurs. In the case of SARS-CoV-1, it was cats. In case of MERS coronavirus, it was camels. And in case of the SARS-CoV-2, it is suspected to be an anteater known as pangolin. There is a publication which also says that it could be that bat is the intermediate host and not the pangolin. Nevertheless, bats and pangolins are involved before the virus spilled over to human beings. Next slide, please. The classification of coronaviruses, they belong to nidovirales, family Coronaviridae, and subfamily Orthocoronavirinae. And there are 4 different genera in Orthocoronavirinae. They are classified as alpha, beta, gamma, and delta. SARS-CoV-2 belongs to beta coronaviruses. In that, there is a subgenus known as sarbecovirus, and SARS-CoV-2 is a member of the subgenus sarbecoviruses. The other beta coronaviruses are OC43, SARS-CoV-1, HKU1, and MERS coronaviruses. Now, this name was given by ICTV, International Committee on Taxonomy of Viruses, in February 2020. Next slide, please. The structure, all of you know, it's one of the most popular cartoons. I think most viewed cartoons. It is spherical, anywhere between 60 to 120 nanometers in diameter. It is enveloped, which is in the cartoon on the left depicted by a blue circle. Embedded in the envelope is the most important protein of the virus, known as the spike protein. This is a pin-shaped protein. It has a head and a stem. The head, the globular portion, is the one that engages with host cells to gain entry, and the other proteins in the envelope are the membrane and the envelope proteins. The virus envelope encases a nucleocapsid, and which carries a single-stranded, positive-stranded RNA as the genome. Next slide, please. This is Hamara DEC SARS-CoV-2, the first virus isolate obtained by National Institute of Virology in February 2020. You can see the nice electron micrograph with the spikes of the proteins on the left and the various intracellular forms of the virus depicted on the right. You can see the nice budding from the cell surface as well. Next slide, please. Going to the genomic organization of SARS-CoV-2, the genome is RNA, it is positive stranded. For those youngsters who do not know what positive stranded is, if the virus genome is introduced into a cell, it will directly go into protein translation. The genome measures approximately 29,600 nucleotides. It has two large open reading frames known as ORF1 and ORF1A and 1B. These two open reading frames code for a polyprotein, and followed by that towards the 3' end are the genes encoding for the structural proteins such as the spike, the envelope, the matrix, and the nucleoprotein. The ORF1 and 2 also code for enzymes such as papain protease and helicase, as well as the RNA dependent RNA polymerase. All the non-structural proteins also come from these two ORFs. Next slide, please. What are the functions of the various proteins? As I said earlier, the spike protein is the most important protein for the virus for its attachment and penetration into host cells, and it is also the most immunogenic protein. So all protective immune responses are directed against this protein. The matrix protein is involved in viral integration. The envelope protein facilitates assembly and envelope formation and budding within the cell when it replicates. The nucleoprotein has a critical role in packaging of virons before it exists. What is more important is there are about 16 non-structural proteins. Several of them we are trying to understand the functions, but more interestingly, at least 3 to 5 non-structural proteins have been shown to have a profound influence on the immune response. NSP1 and 3 are known to inhibit interferon signaling and blocking of host innate immune responses, whereas NSP non-structured protein 3 and 5 greatly promote cytokine expression. This is one of the reasons why you get a cytokine storm from the innate pathway, and NSP16, which is responsible for avoiding the innate immune response, mainly through MDA5 recognition. Next slide, please. The physical and chemical properties of SARS-CoV-2, it is a virus that is stable at lower temperatures and is inactivated within 5 minutes at 70 degrees centigrade, and it's highly sensitive to ultraviolet rays. In terms of chemical stability, being an enveloped virus, it is susceptible to disintegration by lipid solvents, be it ethanol or be it chlorine containing disinfectants and chloroform. Soap is an excellent detergent that dissolves the lipid bilayer and results in SARS-CoV-2 inactivation. A lot has been said about viability of SARS-CoV-2. It remains viable in aerosols for several hours. There are studies which have shown that it is viable for up to 3 hours, as well as it survives on surfaces, more on plastic and stainless steel, where it can survive up to 72 hours, on porous surfaces such as wood and cardboard, it survives for much less time. The masks are used for personal protection. There are different types of masks that have various efficiencies. I think you all know about it. The N95 has the maximum efficiency. Next slide, please. Moving on to the biology of SARS-CoV-2 SARS-CoV-2 virus and its multiplication, I'll cover under 4 slides on attachment, entry, life cycle, and then of course talk about permissiveness to various cell types. Next slide, please. SARS-CoV-2 attaches to human cells using its spike protein. The receptor for the virus is angiotensin converting enzyme 2. This is a physiological molecule involved in blood pressure regulation and the virus attaches there and gets internalized. And there is a serine protease known as TMPRSS2, which actually helps to pull the virus into the cell and facilitating entry. Next slide, please. Once the virus is delivered inside, the virus envelope is destroyed, as well as the nucleoprotein is destroyed by the cellular enzymes. Virus genome is released. Immediately it goes on to the ribosomes and goes into translation because it's a positive sense RNA. It makes several subgenomic uh, copies first of the negative sense, which are then copied to the positive sense, and they go on to make the various proteins, structural proteins such as nucleoprotein, spike, membrane, envelope. And these proteins are assembled on the endoplasmic reticulum membrane, and the non-structural proteins and the matrix and the nucleoprotein ultimately help in assembly. The virus is pinched off the ER and acquires an envelope, and finally it buds out of the cell surface. Next slide, please. Acetyl, the angiotensin converting enzyme 2 is expressed in various parts of the human body. The highest density of expression is in the intestines, followed by the testis and the kidney. There is moderate level of expression in the lungs, the heart, and the blood vessels, as well as in the vessels of the brain. So virtually ACE2 receptors are distributed all over the human body, rendering the human body, various cells in various organs, amenable to virus infection. Next slide, please. If you look at the replication of SARS-CoV-2 in vitro in cell cultures, there have been some elegant publications which I have cited here, one in Emerging Infectious Disease and the other in Lancet. Next slide, please. The summary of this is that this slide shows you the growth curve of SARS-CoV-2 in two different cell lines. One is Vero CCL81 cells. The lines in black show you the kinetics of replication in Vero cells. And the other is human hepatoma cells, or HUH7, and that is depicted in green. In both these cells, virus replication starts very early as 2 hours. It reaches a peak within 24 hours, and then the growth continues on for 72 hours in Vero cells. And in human hepatoma cells, they die, they release large amounts of virus, and then the cells are dead by 72 hours. Next slide, please. Apart from these two cell lines, and these are the plaque characteristics in the Vero cells as well as in the CCL81 cells. And you can see that you get large plaques both on day 2 and day 3 in these cells. Next slide, please. Next slide. Apart from Vero and human hepatoma cells, a number of cell lines are susceptible. It seems to be a very promiscuous virus because it can gain entry into a range of epithelial cells, right from Vero, as I said, to mouse fibroblasts, 3T3, L929, which is a cell line, epithelial cell line of the intestine. Then you can get it in kidney cells, you can get it in Marine Darby kidney cells. Even neuronal cells are susceptible to SARS-CoV-2 infection. There are a series of publications exclusively on neuronal cells. So it's a promiscuous virus, wide variety of cell lines it enters and multiplies. Next slide, please. This is the phylogenetic tree describing where the SARS-CoV-2 is in the entire tree. As you can see, it is far away from the SARS-CoV-1, which is the red alphabets on the top. The SARS-CoV-2 is way down below. It is very closely related genetically to the human bat virus. Because the spike protein of this virus is different from the other coronaviruses, it has been classified separately as a species. Next slide, please. I share with you some of our own experience with molecular epidemiology. Before I go into that, I just want to tell you that SARS-CoV-2 has spilled over from humans to animals as well. There are reports of cats, dogs, and tigers. But the most important report which emerged sometime 2 months ago from Netherlands said that human SARS-CoV-2 infected minks in mink farms in Netherlands, and the cycle was completed again by human beings getting infected from minks. So we know that there is an exchange between human beings and minks in farms in Netherlands. Next slide, please. We had the opportunity to study the SARS-CoV-2 molecular epidemiology in Karnataka during the lockdown period. Before I go into that, let me introduce you to the various clades of SARS-CoV-2 that are present globally today. This is a map showing you the various clades. The S, L, and V on the right, you see as narrow projections or narrow rainbows, are the ones that they are the oldest virus that originated in Wuhan. By February, March, these were replaced with the G clade and the variants of G clade. This classification of clades is based on the GIS-AID classification. As you can see, G has completely taken over from the older clades of S, L, and V. Next slide, please. This is the current global distribution of SARS-CoV-2 clades. The size of the blob tells you the number of genome sequenced. India has sequenced close to 2,000 genomes. We ourselves have sequenced around 110 genomes. And as you can see, in India, all the rainbow colors are there, which means we have the newer genotypes contributing to a majority of the clades in the country, but we still have S, L, and V. These were very early introductions into our country. Next slide, please. Okay, this is a correlation between another classification using pangolin, where the clades were called as A, B, and within B there were subtypes of B.1, B.1.1, B2, B4, B6, and B8. Next slide, please. So people should not get confused. The question we were asking is, what are the lineages that are present in our state? This paper, I'm happy to say, was accepted a week back in PLOS 1, the genomic epidemiology in Karnataka. Next slide, please. We had the opportunity to study 1,578 patients in the state between March 5th and March 21st. And you can see the geographic distribution of the clades, of the positive cases in various districts. Bangalore was the epicenter from where it spread to other parts of Karnataka. The northern districts got it also from domestic travel from other states, especially Mumbai, and the coastal districts. There is an international airport in Mangalore where there were separate introductions. Next slide, please. This slide shows you the 7 lineages that we detected. The panel on the top, A, shows you the various lineages by color codes. Dark blue is A, light blue is B, and so on. So these were clusters that we analyzed and looked for whether one cluster was initiated by one lineage. Almost all of them were, excepting in one cluster, we found 2 lineages, which means there were 2 separate introductions. The panel B down shows the symptomatic versus asymptomatic status in these clusters. The dark circles are symptomatic cases. You can see most of the clusters, the center was a symptomatic patient. And panel C shows you the geographic distribution of the clusters in the various districts. Next slide, please. This is a time course of events that occurred in relation to the cluster and the lineage. Karnataka went into lockdown a week before the national lockdown. And then you see the various clusters that we have described by different color codes. We had a unique cluster, which is the top one in dark green. It's in a place called Nanjangud, close to Mysore, where the cluster analysis and the genomic analysis told us that this was a lineage introduced from Europe, whereas all the light blue ones you see are the clusters that where the main lineage that was seen belonged to the religious congregation in Delhi. Those travelers came to Karnataka and introduced it in various districts. Next slide, please. So this is one of the first studies. Next slide, please. This is one of the first studies to link epidemiological data in relation to transmission along with molecular epidemiology. And such approaches help us understand how introductions occur and how the spreads occur within a state. Next slide, please. The second important lesson with respect to virology is the transmission dynamics that we learned in Karnataka. Next slide, please. I think those of you who attended the morning talk, this would be a repetition. This is a map of heat map of Karnataka during the lockdown period. Red means large number of cases. Green means no cases are very less, and the other colors moderate amount of cases. This is about 3,404 cases. 90% were asymptomatic, 10% were symptomatic. The symptomatic were more in the elderly group with comorbidities, and the symptomatic cases were responsible for a majority of the spread and also sustaining the transmission. Next slide, please. That is explained in this wonderful Rangoli diagram. Click on it, please. Yes. This is a detailed analysis of 46 clusters. Can you go back? Go back one. Each cluster is depicted as a collection of dots. Red means symptomatic case, yellow means asymptomatic, and the center of a cluster is an index case. Majority of the large clusters had symptomatic patients as the index case, and you can also see the size of clusters are bigger when the index case is symptomatic. Transmission occurred both between symptomatic and asymptomatic cases, but if you look at the chains of transmission, they seem to be longer: primary, secondary, tertiary, and so on. Next slide summarizes the arithmetic of this whole analysis that we did. Among the 46 clusters, 32 clusters had symptomatic patient as an index case and 14 patients as an asymptomatic case. The symptomatic patients, 32 of them transmitted to 545, which accounted for 82.7%. The asymptomatics accounted for 17% transmission. The most stunning analysis statistically was the last column of the dispersion index, represented as K value. You can see symptomatics on an average transmit to 14 people, whereas asymptomatic transmit only to 3 people. Very happy to say that this is accepted 2 days ago in Lancet E-Clinical Medicine after several rounds of answering queries to the referees. Next slide, please. A word about the pathogenesis of SARS-CoV-2. Next slide, please. You all know that there are many mechanisms by which this virus produces disease, the most important being by dysregulating the immune response, leading to an aggressive response, cytokine outpour. The other mechanism is by damaging endothelial cells and inducing clot formation. Direct virus-mediated lysis is of very little contribution, and all that occurs in the lung. Next slide, please. The immune response, both adaptive and innate immune responses are important in SARS-CoV-2. You all know a lot about adaptive response. Next slide, please. I thought I'll just touch on one aspect of innate immune response. A very elegant study published in Cell showed that bronchoalveolar lavage fluid collected from SARS-CoV-2 severe patients, as well as those SARS patients due to other viruses, were collected, and they did whole genome sequencing of the cytokine genes, and they found that exclusively in SARS-CoV-2 patients in the severe category, there's a huge upregulation of pro-inflammatory cytokines. So a dysregulated innate immune response is also one of the contributing factors to disease production. Next slide, please. Next slide. Morning, Dr. Ranadeep Guleria beautifully summarized clinical manifestations. The only point I want to make on this slide is that it's a multi-organ, multi-system disease. And today the adage is, if you know SARS, you know medicine, because every possible major system and organ has been damaged by SARS-CoV-2, either directly or indirectly. Next slide, please. We all know the kinetics of SARS-CoV-2 infection. Virus replication occurs for up to a week to 10 days, following which antibodies kick in, and then antibodies persist. We know as of today for between 3 to 6 months in majority of the people. And the first week is up to 2 weeks is the window for detecting viral nucleic acid. Up to a week to 10 days is detection of antigen. Next slide, please. Antibodies have very little diagnostic value. Having said that, this study from Mount Sinai showed that there is a crossover, a window between the 9th and 11th day when the RNA detection is going down and antibody detection is appearing in a brisk manner, so you can detect antibodies. An intelligent combination of these, using these two, can be exploited for diagnosis in tricky patients. Next slide, please. This is one of my last slides. There are many therapeutic approaches for COVID, various stages in virus attachment and replication have been targeted. Having said that, there is no SARS-CoV-2 specific drug available. So we have to rely on repurposing of other drugs, other antivirals like remdesivir, favipiravir. Remdesivir is an Ebola drug. Favipiravir is an anti-H1N1 drug, and ribavirin is a broad antiviral. People have also used immunomodulators. Tocilizumab is an IL-6 inhibitor, whereas the chloroquine was touted as an immunomodulator. And very recently, ivermectin has also been suggested as one of the immunomodulators preventing entry of certain viral proteins transport into the nucleus. Apart from this, indirect therapies like convalescent plasma have had their place, and now randomized control trials says they are not very effective. And vaccines, of course, you just heard Dr. Gagandeep Kang give you an excellent exposition. Next slide, please. In summary, SARS-CoV-2 is a novel virus that was detected in humans a year ago. We have unraveled many aspects of virus biology and transmission in a very short time. That's a great tribute to the technology and the competence of virologists in collaboration with a number of cell biologists and immunologists, epidemiologists, pharmacologists, and so on. But at the end, we know more about the virus than the disease. We are still understanding the disease. In my opinion, SARS-CoV-2 has provided a great opportunity for collaboration, intersector, interdisciplinary, in medicine, in biology, and in chemistry. I think this is a great lesson for the future. Before I end, next slide, please. I would like to thank my team, especially Professor Anita Desai, who has been my colleague for 30 years, and all other my colleagues, Dr. Chitra, who did the genomic sequencing, Dr. Manjunath, Dr. Rita Mani, and all my students who have pitched in during this hour of crisis. Next slide, please. Thank you very much. If there are any questions, I will take it.
Thank you for the presentation, Dr. Ravi. A very holistic approach to the presentation in which you have highlighted regarding the cluster, regarding the cluster analysis, and molecular epidemiology, especially from Karnataka, and regarding the transmission dynamics. Indeed, it has been a very very interesting lecture, and everything has been packaged in a very receptive way and in a very easy to understand way. If there are any questions, I think Pragya would be
Yes,
asking.
ma'am. Thank you, ma'am. Thank you, sir, for this very informative lecture. So there is one question from Dr. Aruna Lok, sir. COVID-19 is an RNA virus where mutation is common. Why mutation in spike is taking so much time?
Well, coronaviruses are genetically far more stable than other RNA viruses, flu viruses. General time for mutation taken is much longer than other RNA viruses. Having said that, in February there was a mutation known as D614G, and that is the G clade that came up and took over the older strains of SARS-CoV-2. But this mutation worked to the advantage of the virus. This D is aspartic acid at amino acid position 614. It got converted to a glycine. What it did to the virus is it made it more permissive. Several cell culture experiments have shown that, within quotes, infectiousness is more with this mutant of the virus. Other than this, there have been no significant mutations in the spike protein. I don't know when it will occur. When I start my viral astrology company, I will answer Arunolog, and maybe in the debate tomorrow. The second question is, more recent papers show there is little cytokine storm in COVID-19. Rather, IL-6 was misused. IL-6 inhibitors were misused in this infection. The answer is yes and no. It is very important to realize that IL-6 is not the only pro-inflammatory cytokine that goes up. There are a number of others that go up. All of them collectively contributed to flooding of the alveoli. That is the primary event in the lung that is responsible. So IL-6 inhibitors, yes, in randomized clinical trials have not stood the test. But generally, steroids, which are known as global immunosuppressors or anti-inflammatory, they are the ones that people are relying on. There's one more question. I can't read it. Can you read it for
Yeah,
me,
sir.
Pragya?
Yes, sir. How long the recovered? Am I audible, sir?
Yes.
How long the recovered of COVID patients are protected by own immune system?
Well, initially we said the answer we do not know. Papers published in the last 1 week have been extremely encouraging. They show that majority of the patients have antibody responses lasting up to 6 months, and a couple of them up to 8 months. And in those where there was no antibody response, specifically they have looked at T cell responses, and that has been very encouraging. So we believe that immune responses will last, but only time can ultimately answer that question. If you look at the history of other related coronaviruses, immunity does last long, like many other RNA viral infections. So I believe the answer is yes, the immunity is going to last. One point I forgot to mention in my talk, I would like, many people ask me on reinfection. There is no clear cut definition of reinfection. But having said that, there is a consensus that when do you say a person is reinfected? The specimen collected during the initial episode should be positive. After that, when the patient has recovered, there should be a negative RNA report. Later on, 4 weeks or later, if there is a second episode and there is a second specimen, there should be a positive evidence of molecular typing, especially genome sequencing, is very important tool. The CDC protocol very clearly says you should use the genome sequencing, whole genome sequencing, to definitely show that there are differences in the nucleotides between the first episode and the second episode. Over.
Thank you, sir. Sir, I would like to add one more question. How far is this clade variation going to affect the sensitivity and specificity of PCR platform?
The sensitivity and specificity of PCR detection is not going to be greatly affected because, we rely on multiple targets. And all these primers are designed in the conserved regions of the genome, and we have not seen uh, mutations. See, every mutation in the nucleotide doesn't translate into an amino acid change, and every mutation, if it is a single point mutation, it will not greatly affect the sensitivity of the PCR. Over.
Thank you so much, sir. I think there are no more questions, ma'am. Dr. Anita, ma'am.
Yeah, thank you, Dr. Ravi, for such an excellent presentation and such good answers to all the queries. I'm sure all the delegates have really enjoyed the session because the core of virology is the basics. And the molecular is the applied part, of course, which is being used by everyone. And I'm really happy to see the core being covered by a very dedicated virologist. Thank you for this session.